So today I would like to talk about my PhD project, which I did in Guillaume Dis lab at the FMI in Basel. And a lot of the things I will say also in my, especially my introduction, will sound very familiar after the last talk you just heard. Um, but we will look at this whole question of protein function um, at a kind of different angle and looking at a different kind of protein function. So also in the DIS lab, our driving question of our research is that of how genotypes and phenotypes are related. And the central dogma clearly states that DNA is translated into RNA sequence, which is then translated into protein sequence and protein structure. However, how we get from this protein structure to a molecular function and ultimately a phenotype is somewhat less well understood. We do know, though, that a major that something that drives molecular function is functions are molecular interactions, and especially protein-protein interactions, since proteins perform most biochemical functions within a cell, and they do so by interacting with one another. The inside of a cell, two proteins in a cell do not exist in isolation and can easily find each other when they want to interact with one another. But cells are more these very crowded, highly concentrated environments with millions of protein molecules that need to find each other. Therefore, the interactions need to be highly specific. And the protein-protein interaction specificity is what's encoded in DNA sequence. So mutations can actually alter these specificities, thereby perturb molecular interactions and thereby perturb molecular functions and even cause diseases. As mentioned before, since the launch of AlphaFold, we have made great progresses in predicting protein function, especially folding and also protein-protein interactions, just because AlphaFold is this very powerful neural network trained on an immense data set of protein structures. However, there are still limitations. For example, when it comes to making quantitative predictions about, for example, interaction strength, or predicting the effects of mutations on protein-protein interactions. Therefore, the goal in our lab was to develop new experimental and analytical approaches to allow large-scale data of how mutations perturb protein-protein interactions, which can then be used to train quantitative models that are able to predict protein-protein interaction affinity and specificity from sequence. But how large and extensive do these data sets actually have to be? And to answer this question, we first need to understand how complex the genetic architecture of protein function, or rather protein-protein interaction, really is. And there's on the one hand the possibility that genetic architecture of protein function is simple, so that individual residues only contribute additively to biological function, or differently said, the same mutation occurring in a different genetic background or context has largely the same effect. On the other hand, protein function can be very complex, which means we observe pervasive epistasis of pairwise and even higher order, where the same mutation in a different context has a very different effect on a protein function, or in our case, protein-protein interaction. And the most recent studies looking at different deep mutagenesis data sets have shown that actually it seems that epistasis is not so pervasive as was thought previously, and that models that only look at first order mutations actually can very accurately predict the effect of mutations, and only in isolated cases taking into account higher order epistasis actually improves our prediction. So it looks like the general genetic architecture of proteins seems to be more on the simple side. However, what we need to take into consideration here is that all these data sets that have been studied only looked at mutations in single proteins and the mutation effect basically in their local neighborhood. But what actually happens if we consider a whole protein family or protein domain family, where we have a very widely sampled sequence space, actually, because these different family members have rather distant distant sequences, but they have very similar functions, as, for example, is the case for the family of the basic leucine zipper domains, or BZIPs, that are quite different in sequence, but they all mediate these dimerization via coiled coils. So therefore, our approach was to perform now the deep mutational scanning of all members of the BZIP family, and then compare the effect of the same mutation in the different family members, so in different contexts, and learn how this context actually influences the mutation effect. So quick introduction on the BZIPs. There are 54 in humans. They occur in transcription factor and mediate homo and heterodimerization. And their interface is relatively simple. They're alpha helices that can be split in a 27 amino acid long DNA binding domain, a 35 amino acid long zipper domain, which can be further subdivided into 5 haptids that each consist of 7 amino acids. And these 7 amino acids always take the same position relative to the opposite molecule in one of these dimers. A and D form a hydrophobic core, E and G form salt bridges to stabilize the dimer, and B, C, and F are solvent exposed and often polar. The BZIP network is very modular, which means that we observe a high variability in specificity, where subfamilies of BZIPs interact strongly together. And in between these subfamilies, as indicated by this yellowish color here, we see low levels of interaction or crosstalk. So our aim to now understand the genetic complexity or genetic architecture of this network was to perform a deep mutational scanning of the entire human BZIP family, where we basically create every possible single amino acid mutation in every one of the 54 BZIPs, and then measure the interaction of these mutants with all 54 wild-type BZIP, so that actually for each pixel in this all-by-all wild-type heat map, we make one of these deep mutational scanning heat maps where we see each amino acid in the rows at each position within the zipper domain in the columns, and the color indicates then a binding score that we measured for the given mutant. The assay that we're using is the same one that Tony just presented, based on the split dihydrofolate reductase, but we use it in a slightly different way to measure protein-protein interaction. So we're using this methotrexate insensitive DHFR variant that's split in half, and each half is used to tag one of 2 proteins of interest that we want to measure the interaction of. These proteins are then expressed in yeast cells, and in case of interaction, DHFR is reconstituted and active in presence of the drug methotrexate, which allows cell growth in a rate proportional to the amount of reconstituted DHFR activity. And this quantitative nature of the assay then allows us to use this in a pooled deep mutational scanning format. So we can clone plasmid libraries where we express different combinations of proteins from the same plasmid, transform this plasmid library into yeast cells so that every yeast cell expresses one pair of tagged proteins. And we then let them grow in a pooled culture under selective conditions, so that we only enrich for cells expressing interacting proteins. And by comparing input and output cultures, or comparing the frequencies of plasmids in input-output cultures using deep sequencing, we can then infer interaction fitness enrichment scores for the given pairs of proteins. We do this in a library screening setup, where from the plasmid we express on one side our BZIP mutant library of all 38,000 mutants in all 54 BZIPs, and on the other side the library of 54 wild-type BZIPs. But before we did this massive venture, basically, we decided to start with a proof of concept where we did this only for the June as one of the most prominent members of the BZAP family. So we mutated every possible single amino acid in June and then measured the interaction with all 54 wild-type BZAPs. First of all, we looked at the binding score of the 54 BZAPs with wild-type June and mostly recapitulated what's already known. So there are some BZAPs that can interact with June at different strength, and like 1/3 of them, and the rest, 2/3 remaining ones do not interact with wild-type June. And for the mutants, we then calculated binding scores relative to the wild-type interaction, where blue color indicates a reduced interaction compared to wild-type and red an increased one, and plotted this on this huge heat map where the rows represent the interaction partner and the columns represent the amino acid so the amino acids at a given position. For example, haptid 1 position A here at the very first columns, and then what effect on the interaction the given mutation had. And what's striking first is that most mutations seem to have a similar effect across the partners, which is indicated by these blue vertical lines. But additionally, we also have these few specific effects where the effect we're observing seems to be only present in a specific set of partners. If we plot the mutation effects of different BZAPs against each other, we also observe that they follow a trend, but this trend seems to be nonlinear. And so there seems to be a similar effect, but they're not linearly related. And this lack of linearity we can actually explain by thermodynamics. So for example, if we look at the blue and the yellow mutation, and we assume that they have the same effect no matter if June interacts with JDP2 or with FOS. So in the case of JDP2, where the wild-type interaction between June and JDP2 is already really strong, the mutation that has a certain delta G will result in only a small measurable difference in how many dimers can be formed, because we're already almost all proteins in the cell are in a dimer. So even if the mutation increases the interaction strength, we will not form much more dimers and will therefore not see a strong change in our fitness score. On the other hand, for FOS, if the mutation has the same delta G, but our FOS-June interaction is not as strong to begin with, we can actually get a much larger difference in fitness effect here. So, so this is why we don't see this nonlinear relationship here. Additionally, we also have a few points that fall off this trend, and these are the specific effects that I mentioned earlier. So since we have now these different effects, the specific and general effects, we also wanted to kind of formally quantify this and really figure out what the energy changes here are. And therefore, we use this same model that Tony presented earlier, which is MOCHI, to fit delta G's. And here, in this case, we assume a two-state equilibrium of BZIPs of unfolded unbound monomers and folded and bound dimers. And MOCHI then, under the assumption that all mutations are additive, infers these delta G's for the different mutations that represent the delta G that is shared across all partners. And when we plot these predicted binding scores versus observed binding scores, we see that they correlate very well, which confirms that indeed most mutations have a shared effect across all partners. We also find a very high correlation of these inferred delta G's with in vitro data of BZIP interaction strength that was already published before. If we look at these global energy changes that are shared across all partners and how they are, what kind of characteristics of June they actually affect, we see that there are There are characteristics like helical propensity, hydrophobicity, all characteristics that are related to June's general ability to form a cold coil with another B-zip. And we call this zipper propensity. In addition, I already also mentioned these specific effects though, and these are now residuals that fall off this curve, of this Mochi curve between this trend between delta delta G's and binding scores. So basically, if the measured mutant and the predicted mutant are not the same, we have a residual, and this is what we call the specific effect, something that we only observe between June and a specific partner and not with all partners. And if we plot now our heat map only with these residuals, with these specific effects, we can actually identify determinants of specificity for June. And with we have positive determinants of specificity. These are positions in June that in their wild type variant are important to maintain an interaction with a given partner, because if we mutate them, the interaction is lost. We also have negative determinants of specificity that in their wild type variant are important for preventing a given interaction of June with a partner, because if we mutate them, we can increase or induce an interaction here. And we also have dual determinants of specificity, where even within the same position, mutations can both increase and decrease interaction specificity. For example, this position 3A, where June has an asparagine, and June does interact with Phos in the wild type, and Phos has a lysine. But if we would mutate this lysine to a valine, the interaction between Phos and June would be a lot stronger. So the lysine here, the asparagine here is actually not ideal. However, if we would mutate to a valine, we would also induce a novel interaction between June and the MAF subfamily, which is unwanted. So we think that these positions kind of function as these trade-off positions where the network is really fine-tuned. So it could be optimized, but optimization would result in unwanted crosstalk. So to summarize what I've shown you in this first part, single mutations in June have additive effects and are mostly independent of context, meaning they affect all partners similarly. And this is a global non-specific component of binding affinity that we call zipper propensity. The specific component of the mutation effects defines binding specificity to different subfamilies and depends on the partner's sequence context. To summarize, this kind of supports what has been previously shown, that the genetic architecture of individual proteins is relatively simple and mostly defined by these additive non-specific contributions to affinity, and we only fine-tune it with specific effects. But what is now actually the full story? What if we look at all 54 wild-type BZIP at the full deep mutational scanning? So we did this, as I mentioned earlier, and as a result, we get this massive heat map of 2 million quantitative measurements of PPI. And this basically represents 3,000 deep mutational scans done in one, which is more than the entire MAVE database at the moment. This data set is super versatile, and we can actually look at many different characteristics of BZIP biology. For example, we can already, just looking at the heat maps, see some general trends that are not very surprising, but kind of reassuring that what we're doing is actually working. For example, mutations at A and D positions, at the core positions, are most disruptive of interaction. Proline mutations, of course, which break the helices, also disrupt dimer formation, or same for stop codons. But we also found some very interesting observations. For example, single mutations can form a completely orthogonal BZIP, as it is the case for the SCRAP5 mutations. So here on the lower part of the graph, you see the interaction partners of wild-type CRAP5. And if we mutate position 3A to an isoleucine, all these wild-type interactions are lost, and we gain these upper interactions here with the Bach subfamily, and this mutated CRAP5 can now also homodimerize. And this is not an isolated case. We actually find hundreds of examples for this. And we find this very interesting because this might actually be a possible explanation for the evolutionary success of the BZIP family, because it is a quite big family that expanded to 54 members, maybe because it is so easy for them to refunctionalize or form, or just like basically a single evolutionary step, a single mutation can give rise to a protein with a new function, as in a new interaction network. Also here, of course, we were very interested in seeing what happens if we run mochi on it. And we did the same as for June here, but for each BZIP individually. And we also were able to determine these, to find these determinants of specificity. And it was interesting because we see similar determinants across the BZIPs, but they are basically used in different combinations. And for example, if we see BZIPs from the same subfamily, their determinant patterns look very similar. If they're more distant, they use kind of different combinations. So these determinants of specificity are kind of shared across the network, but then used in different combinations in order to fine-tune the network. When we then look at the total delta G, so the total mutation effect of all the mutations, and plot their correlations, we see that they are very similar in related BZIPs that come from the same subfamily as these two from the MAF subfamily. But if they're more distant, like for example, between MAF and BATF subfamily, the correlation is extremely poor. So here the mutation effects are very not similar. So they are very different. And if we plot this for the entire network, the correlation between the mutation effects, we see that this really nicely recapitulates the family architecture of the BZIPs. So we have the subfamilies clustering together with high correlation, and in between the subfamilies, we have lower correlations. So basically what this indicates is that the genetic architecture of the whole BZIP family whole BZIP family function is more complex than we originally anticipated. So same mutations in different contexts actually have different effects. Yes. But is this now a reason to panic? Does this mean we cannot build good models to predict the effect of mutations for entire protein families? And the answer is no, because we were able, with collaborators from FMI, to to build a relatively simple convolutional network that can predict the interaction scores very well from sequence. And interestingly, we used different strategies for data splitting here, and the model performed most poorly when we left out entire proteins out of the training set. So basically, when we masked parts of the sequence landscape that we had sampled, then really the network kind of had a harder time predicting the mutation effects, kind of reassuring the strategy of sampling as wide of a sequence space as possible for protein function. We took this one step further and used our neural network to predict synthetic BZIPs as competitive inhibitors for 8 chosen targets that come from 8 different BZIP subfamilies. And the network basically predicted these binding scores or enrichment scores for the 10 binders that are shown in the columns. And we ran a screen to test these, and our measured enrichment scores look really similar to the predicted one, which is very promising as well. And interestingly here, our predicted binders are quite distant from our BZIPs. So they are outside of the sample sequence space, and still the network performs well. So to summarize this, when we do wider sampling of a sequence space, we observe that the genetic architecture of the biological function of protein-protein interactions seems to be more complex than if we only consider local neighborhoods of proteins. However, we can still use relatively simple deep learning models that are able to capture the sequence function relationship and make accurate predictions. I should mention that BZIPs are relatively simple protein domains. They are convolutional in nature, so maybe that is why this convolutional model also performs so well in predicting them. So now outlook or the goal of the lab is, of course, to apply this approach to more complex protein domain families and check if we observe similar phenomena. And currently in the pipeline is, for example, the SH3 domain family and even larger deep mutational scan here. So basically, to even zoom out of our landscape even further, the idea is now to really to get towards this general model of protein-protein interaction. We need to try to sample as much sequence space as we can of different protein domains and understand how the genetic context really influences these protein function. And with this, I'm at the end of my talk, so thank you very much for your attention and for joining. I want to thank my lab, my PhD lab at the FMI, especially Guillaume for supervising my PhD. It was a great time, and I obviously miss the lab a lot. And also thanks to all of our collaborating labs at FMI and our great facilities there. It was a super nice experience to work there. And thank you also to Ben and André for the nice collaboration on the June project, which was also very great. And I'm supposed to mention here that the DIS Lab is looking for a postdoctoral fellow. So if you're interested in these kinds of projects, feel free to reach out to Guillaume. And I can highly recommend working there, of course. Yes. Thank you.
All right. So thanks, Alex, for a really great talk. I think in the interest of time, we'll answer one question from the chat. And then if anybody else has questions, I encourage you to reach out to Alex directly. So Justin asks, have you compared the results of your deep mutational scan and simple convolutional network to general protein interaction prediction tools like AlphaFold Multimer?
Yes, we have tried to use AlphaFold to predict this. I didn't do it myself, and it's also quite some time ago, but it did not work well. So it could not predict the point mutation effect. That much I remember, but I would have to go back and look in more detail. So whoever asked the question, if you're interested in details, you can write me an email and I will look back into it.
All right. Thanks to both of our speakers and to everyone who joined us today. And we'll look forward to catching up next month and hearing from more great talks. Thanks, everyone.
Thank
Thank
you.
you.